everyone, welcome to our channel Biology for All. In this video, I'm going to explain the stages of mitosis. Mitosis is an equational division which has 4 phases. Let's see the first phase. The first phase is prophase. Now, prophase has certain identifying features. The first one here that we can see is the movement of the centrosome. The centrosome has already split during interphase stage, and during prophase they start moving towards the poles. As they start moving towards the poles, they create the spindle fibers in between them. The other feature of the prophase stage is the disappearing nuclear membrane. So these nuclear membrane starts to disintegrate. Inside that we can see the presence of chromosomes. Chromosomes have condensed and become thick and dark, and it is easily stained during this phase. In fact, we can see it in the form of the sister chromatids also. So these are the features that differentiate the prophase stage in mitosis. The next stage is metaphase. Now, as the nuclear membrane has disappeared, the chromosomes now start to move towards the center of the cell or towards the equator. The centrosomes have already moved towards the poles, thus creating the spindle fibers. So from the centrosomes, that is the centrioles inside, there are rays that come out in the form of spindle fibers, which are the ones that are between the two centrosomes, and the ones that radiate and do not join with the other are called as the astral rays. So the centriole and the astral rays together form the aster. There are fibers which extend from one pole to the other. These are called as continuous fibers or polar fibers. The other fibers or the other spindle microtubules are the ones which are attached to the chromosomes. So these are called as the kinetochore fibers, as they attach to the kinetochores on the centromere. So these are called as kinetochore microtubules, or they are called as discontinuous microtubules. The metaphase stage shows the presence of chromosomes on the metaphase plate. So metaphase plate or the equatorial plate, or it is simply called as the equator. So the chromosomes come and align on this equator or equatorial plate, such a way that the centromeres are on the equator and the arms are facing towards the poles. So this is the centromere which is on the equator and the arms that are facing towards the poles. Now here this is one chromosome. So here the sister chromatids are attached at the centromere. So that blue structure that we see here is the centromere, and next to the centromere there are proteins to which the spindle fibers attach. So the proteins on which the spindle fibers attach is called as the kinetochore. So that is the kinetochore. The part on to which the chromatids are attached are called as a centromere, and the spindle fibers are the ones which attach to the kinetochore. So this is how the arrangement is there in metaphase. The next stage is the anaphase. Here you can see that the cell is slightly elongated as compared to the other two stages, and the chromosomes have now split along the centromere and they have started moving towards the poles, or they are being pulled. So here the kinetochore fibers, they undergo depolymerization and they shorten in length. So as they shorten in length, they pull the chromatids towards the poles with their centromere. The polar fibers undergo polymerization and start stretching, unlike the kinetochore fibers which are shortening. So This results, that is the shortening of the kinetochore fibers, result in splitting of the sister chromatids. Thus, now these are called as the daughter chromosomes. So, the sister chromatids split to form the daughter chromosomes. Here in between them, there develops a fiber that is called as the interzonal fibers. So, this is the stage that is the anaphase, which is the shortest stage in the mitotic division. The last stage of mitosis is the telophase stage. Now, the telophase stage marks the completion of karyokinesis or the nuclear division. That is, the nucleus have now split and formed two nuclei. Now, telophase is also called as reverse prophase, as we can see all the structures that were disappeared in the prophase stage return back or reform back. So, the centrosomes are present on each pole of the cell. So, there are two centrosomes now. The nuclear membrane is starting to reform, and the chromosomes, which are now inside the nuclear membrane, start to decondense to become chromatin. In between, they show the constriction, which is called as a cleavage furrow, that then leads to cytokinesis. So, this completes the mitotic division.